So our second video in the membranes series, part 2, IB 2.4, in this one we're going to look specifically more at diffusion osmosis, passive active transport. In the previous video we covered what makes up the plasma membrane and some of the functions of membrane proteins. So let's first start by talking about what diffusion is, and hopefully you remember this as well from biology. Diffusion is a passive, which means that there's no energy required. It's a passive movement of particles from a region of high concentration to low concentration. And so a great example of this process of diffusion is if you have a small beaker and you fill it up with some water, and if you add a drop of Kool-Aid to that water, um, that Kool-Aid is very concentrated at where it contacts or touches the water. And eventually, if you let it sit long enough, it's going to spread out. And so it's moving from a region of high concentration initially to a region of low concentration. And eventually, that's going to continue, and that Kool-Aid is going to spread out until balance or homeostasis is met within the liquid. You could also call that equilibrium. So here's a nice image that kind of shows us what's happening. We've got time here, so this would be the start, and then time progresses. And we've got a membrane, cell membrane in this example. And so we have a lot of particles on this side of the cell membrane. And extracellular space, so outside of the cell, internal space, intercellular space, so inside the cell. As time progresses, these particles are going to move across this membrane from a region of high concentration to a region of low concentration. And this is going to continue until there's a nice balance, until there's a balance or equilibrium on both sides of the cell membrane. This is diffusion. It's just particles moving across the membrane from a region of high concentration to low concentration until equilibrium is met. Osmosis is very similar. It's also passive, so there's no energy required, and it's the passive movement of specifically water molecules. So in diffusion, we can be talking about any different types of molecules. Osmosis is very specifically pertaining to water molecules. And it's the movement of water molecules across a partially permeable membrane from a region of lower solute, like a solid, concentration to a region of higher solute concentration. And so what that looks like is, let's say we have a vial here, or a beaker, or a glass, and we have a semi-permeable or partially permeable membrane that divides the beaker in half. And on one side we have lots of sugar. We have very concentrated sugar solution, and there's some water. And on the right side we have a diluted sugar solution. So in this situation, or in this example, our solute is the sugar. That's the solid. Our solvent, what is dissolving or going to dissolve the solute, is our water. And so we have a very high solute concentration on this side and a low solute concentration on our right side. And as osmosis progresses and as time goes on, we're going to see more water. These little blue dots here are representing water. We're going to see water actually move into the region that has a higher solute concentration. And this is going to continue over time, just like diffusion, until we reach a state of equilibrium. And so now our concentration of water to sugar, or our concentration of solute, the sugar, to our solvent, the water, is going to be equal on both sides of this partially permeable membrane. And this is going to continue, and this is going to occur without energy. So the important thing to remember with both diffusion and osmosis is energy is not required. Some similarities and differences between the two. Diffusion is the movement of solutes, particles, like sugar. In this situation, it's not necessarily, or it's not water that's moving. And a membrane is not necessary for diffusion to occur. Osmosis is the movement of water molecules specifically, and a semi-permeable membrane is required. Okay, so those are how the two are different. They're similar in that they are both passive transport, meaning that they don't require energy. And in both cases, or both situations, it's the movement of molecules down or against the concentration gradient. And that concentration gradient, what that means is we're moving from an area of high concentration to an area of low concentration. So we're moving from an area where there's a lot of something to an area where there's very little of something. And again, that occurs until equilibrium or balance point is met between the two. In looking at passive transport, there's a couple different types. Simple diffusion is basically just diffusion. It's the movement of molecules across semi-permeable membrane until homeostasis is met. And again, it requires no energy. So here, this is an example kind of like what I described earlier. This one's actually using food coloring, which is another great way to observe simple diffusion. You drop that food coloring into the liquid. There's a region of very high concentration where it initially lands, and then eventually it's going to spread out. Kind of a cool lab that you can do is to test the rate of diffusion in different types or different temperatures of liquids. You can actually see a difference between hot and cold water. Facilitated diffusion is similar, but it's the movement of polar molecules or ions. It's moving across a lipid bilayer with the help of a protein. So oftentimes, polar molecules or ions or sometimes larger molecules, they're not able to move across the membrane without some help. And in this case, a protein can actually help this movement, and so we call this facilitated diffusion. We're still moving from an area of high concentration to low concentration, so it doesn't require any energy. And here's a nice image that shows or describes how this actually works. Our first one is a protein channel, and it's kind of like a tunnel. Basically, we have an area of high concentration outside of the cell, and through this protein, our molecules or our ions are moving across the membrane through this protein channel. The other option, or the other possibility, is a protein carrier, and in this case, the shape of the protein is actually changed. Again, we're still moving from an area of high concentration to low concentration, so no energy is required. And in this case, we have instead of a channel, a protein channel or a tunnel, we have a carrier protein that actually changes the shape of the protein in order for the molecule to be moved across the membrane. Active transport is very different from passive transport in that it requires energy. And so what's happening in active transport is we're moving solutes against their concentration gradient through a carrier protein. So this requires energy, and in essence we're moving molecules or solutes from an area of low concentration to high concentration. So it's opposite of passive transport. Again, passive transport is moving molecules from high concentration to low concentration. Active transport is the complete opposite. We're moving from areas of low concentration to high concentration. And in order to do that within the cell, it requires energy. And this allows cells to maintain their internal concentrations of solutes that differ from the external environment. So really what this means is it allows the cells to pump things across the membrane in order to maintain their internal concentrations. And I've got an example of that, and that is the sodium-potassium pump. And so in this case, sodium is pumped out of the cell and potassium is pumped into the cell. And they're both being moved against their concentration gradients. But this is an important process in the cell because the cell needs that potassium in order to carry out different functions. And so in order for that to come into the cell, sodium has to be pumped out. And so here's a nice diagram that explains and shows us what's happening. We've got sodium binding sites and phosphorylation by ATP. So ATP comes along and it binds to our carrier protein here. We've got sodium that's already been put into our carrier protein. And as a phosphate is released from that ATP, so phosphorylation causes the protein to change its conformation, expelling or releasing the sodium to the outside. So ATP loses a phosphorus and so it becomes ADP, and that causes the protein to actually change its shape and it releases the sodium out of the cell. And after that happens, potassium is able to bind to that or be held by that carrier protein, and that portion releases the phosphate. When that phosphate's released, the carrier protein again changes its shape so that the potassium can be released inside the cell, and then basically the process continues. And so what's happening here is we're using ATP, we're using energy, and that energy is driving the conformational change of the carrier protein, and it's allowing sodium to be pumped out of the cell and allows the potassium to be pumped into the cell. And again, this is they're both being moved against their concentration gradient. So within the cell, there's lots of sodium. Outside of the cell, there's lots of sodium, but we're pumping it out, and potassium is being pumped in. There's also transport across the plasma membrane by two different methods called endo and exocytosis. Exocytosis is vesicles or molecules exiting the cell. And in this case, we have a fusion of a vesicle with the plasma membrane. Basically what happens is the vesicle from a Golgi apparatus moves to the plasma membrane. The membrane of the vesicle and the plasma membrane fuse, and then the contents of the vesicle are released outside of the cell. Again, exocytosis is exiting the cell. Endocytosis then is just the opposite, and that's bringing particles or molecules or whatever it may be inside or into the cell. And so this is the reverse of exocytosis. Cell takes in molecules by forming new vesicles from the plasma membrane. So a small portion of that plasma membrane is actually going to kind of pinch off after forming around the molecules that are being brought into the cell, and then it's released into the cell, goes to the Golgi apparatus for further processing. A small portion of the plasma membrane pinches inward. And so here's a nice diagram that actually shows us how this works. Here's our Golgi apparatus. The white portion is within the cell. The kind of gray portion here is outside of the cell. And so we have both scenarios here. A small portion of the Golgi apparatus with vesicle is pinches off from the Golgi apparatus and it's moving towards the plasma membrane. The membrane of the vesicle fuses with the plasma membrane here. The vesicle opens up and it releases that content out into the cell. Okay. Same situation here. We have some molecules that are being released outside of the cell by or through some signal transduction. So there's some sort of hormone, neurotransmitter, something is telling a binding site, a hormone binding site, that this needs to occur, and so the Golgi apparatus releases this vesicle that contains these molecules, fuses with the plasma membrane, and releases outside of the cell. Both of these are exocytosis. Endocytosis is basically the same example, and I have a nice diagram that shows us an example of that. So this is a good medical example of how this endocytosis could be used. Here's our plasma membrane. This is basically the phospholipid bilayer. There are regions both of polar and nonpolar, as we've discussed, water or aqueous environments on either side, our hydrophobic region in the middle. And so these phospholipids, as we discussed, close, or they form some sort of sphere that's called a liposome when mixed with water. And so the hydrophobic portion is protected because on the external and the internal is all the hydrophilic regions. And this is actually one way that we can use this process to deliver drugs to human cells. If we have a drug right here in the middle of a vesicle, it's surrounded by a phospholipid bilayer, that portion can be fused with the plasma membrane to release that drug inside of the cell. And so this is just one medical example of how this process. So the last portion that we're going to look at is the fluidity of the membrane, or how the membrane can move. And we've previously discussed the fluid mosaic model. Fluid meaning that the plasma membrane is able to bend and flex and it can change its shapes, kind of like water. Mosaic meaning that it's composed of multiple different types of molecules, phospholipids, proteins, cholesterols, carbohydrates. And then model meaning that it's a representation for a real item. Phospholipids can move freely and they're able to bend and flex. And this is a nice video that kind of just shows us or describes how this happens in a real example. This is obviously being viewed under a microscope.
To demonstrate the fluidity of the lipid bilayer, a piece of the plasma membrane of this neuronal cell is pulled out with laser tweezers. Remarkably, moving this membrane tubule rapidly back and forth does not rupture the plasma membrane, which flows quickly to adapt to the mechanical distortion.
And so really what that's showing us is that phospholipids can really move freely. Although the phospholipids have weak attractions between each other, the combined attraction creates a very strong membrane. And that's what we saw in this video here, is by actually kind of pulling on the phospholipid bilayer right there, rather than breaking, it's able to move back and forth. And so that's what the fluidity of the membrane kind of represents. And so that wraps up our membrane discussion for IB. To finish up the unit, we'll talk about cell division.